Okay, let's unpack this. Imagine having like a scientific flashlight,
Hmm.
one that lets you see these invisible conversations happening inside our cells
Right.
with incredible clarity and speed. That's really what we're diving into today, this revolutionary technology called NanoBiT.
Yeah, NanoBiT.
Our mission is basically to explore how this really ingenious tool is, well, changing how we understand cellular communication.
Fundamentally changing it.
Yeah, from, how basic life forms manage metabolism
Mm.
to the really complex ways our bodies fight disease, even how viruses get into our cells.
This huge range.
So get ready for some, I think, truly illuminating insights here.
And what's really, I think, transformative about NanoBiT is how it kind of bridges that gap
Gap between what?
between, understanding these fundamental biological processes
Okay.
and then accelerating actual advancements in areas like, say, drug discovery.
Right, the practical side.
Exactly. It lets researchers watch these dynamic interactions in real time
In the cell.
in their native cellular environment, yeah. Providing insights that were just, well, incredibly challenging, maybe even impossible before with the older methods.
So this NanoBiT, it sounds powerful. Can you kind of walk us through the core idea? How does it actually work?
Sure. So at its heart, NanoBiT is quite an elegant adaptation of a natural thing.
Okay.
The name stands for Nanoluke Binary Technology.
Binary, meaning two parts.
Exactly. It uses an engineered enzyme, a luciferase called Nanoluke.
Luciferase like fireflies.
Close.
Mm.
This one originally comes from a deep-sea shrimp, known for really bright bioluminescence. So think of it like a tiny molecular sensor system. It lights up when specific things happen.
A molecular sensor.
Okay.
How does that tell you about proteins interacting inside a cell then?
Right. Okay, so here's where it gets really neat.
Uh-huh.
The Nanoluke enzyme, they split it into two distinct pieces.
Okay.
There's a larger bit called large bit, about 18 kilodaltons,
Right.
and a much, much smaller peptide called small bit, only 1.3 kilodaltons. Very tiny. And crucially, these two pieces, on their own, they have a very weak attraction for each other.
So they don't just stick together normally.
Exactly. Very low affinity. So they don't spontaneously reassemble, and you get very little background light. Minimal signal.
Okay, big
But-
kids.
When you take each of these fragments and fuse them, attach them to two different proteins you want to study,
Your proteins of interest.
create your proteins of interest. And if those two proteins interact inside the cell,
Uh-huh.
then the large bit and the small bit fragments, they get brought really close together.
Because the proteins they're attached to are interacting.
Exactly. This precise bringing together reconstitutes the active luciferase enzyme.
Okay.
And instantly, boom, you get a bright, measurable light signal.
So the light itself is the signal. It's the readout saying these two proteins just interacted. That's remarkably precise.
It is. And this method, it gives you just unprecedented sensitivity and a really high signal-to-background ratio.
Better than the old ways.
Oh, much better in many cases. Unlike lots of older techniques where you might have to, say, stick one protein onto a surface.
Yeah, immobilize it.
Yeah, immobilize it, which can actually, mess with its natural behavior or how accessible it is.
Sure.
NanoBiT lets you measure interactions with the proteins just freely floating around in solution, much closer to how they really are in the cell.
More natural state.
Exactly.
Yeah.
Plus, that extremely low natural attraction between the large and small BT pieces,
Yeah.
that means the light signal you see is a true reflection of the actual protein interaction happening. It's not just the fragments bumping into each other randomly.
Got it. And that low background signal you mentioned, how does that help? Especially for measuring things quantitatively?
Oh, that low background is absolutely critical. It makes the signal exceptionally clear, really reliable,
Okay.
which lets you precisely detect and measure even really weak interactions, or ones that happen very fast, transient ones.
Ah, the fleeting ones.
Exactly. And this is a game changer because, honestly, so many vital biological interactions are fleeting or low affinity.
Right.
They were just incredibly difficult to capture and measure accurately with the traditional methods.
Okay. All right. Let's take this incredible tech and look at the first big application from our sources, understanding these things called PII proteins.
Ah, yes. PI.
They're described as ubiquitous in nature.
Especially in microbes, right? Bacteria, cyanobacteria.
That's right. Very widespread.
What makes them so important? What's their deal?
Well, you can think of PII proteins as the cell's metabolic watchdogs, basically.
Sentinels. Okay.
Yeah, sentinels. Their main job is to constantly sense the cell's nutritional state, its energy status,
Oh.
by binding to key status recorder molecules, things like ATP and ADP.
Energy currency.
Exactly, tells you about energy levels. And also to oxyglutarate, which is a signal for the carbon-nitrogen balance in the cell.
Okay.
And depending on which molecule is bound, the PII protein actually changes its shape, subtly but significantly.
Particularly in these flexible regions they have called T-loops.
T-loops.
Yeah, and these T-loops act like versatile docking sites, interaction modules,
Right.
allowing PII to then interact with and change the activity of loads of different target proteins. It's essentially reading out the metabolic information and coordinating what the cell does next.
So they're totally essential for a cell to know what's going on with its resources. But why was studying these interactions, especially the dynamics, in real time, such a challenge before NanoBiT came along?
Well, the traditional methods, they were powerful, definitely, but they often hit roadblocks with PII,
Like what?
like SBR, surface plasmon resonance,
Okay.
and BLI, biolayer interferometry. Both usually measure binding by seeing how light changes when molecules stick to a sensor surface.
Right, the immobilization problem again?
Exactly. You have to stick one partner down. And for really dynamic proteins like PII, pinning them down can actually restrict their natural shape changes, their movement.
Might change how they behave.
It can. There was one study that found just sticking PII down using a tag on its end actually messed up how it responded to ATP and 2-oxyglutarate.
Wow. Okay.
And then there's FRT analysis, first to resonance energy transfer.
Right, using fluorescence.
Yeah, powerful. But FRT often means fusing pretty big fluorescent proteins onto your targets, and it can be really sensitive to background fluorescence, especially trying to do it in live cells.
Gotcha. So NanoBiT steps in, it avoids those issues. What were some of the really cool new things it showed about PII interactions, especially in those complicated networks?
Oh, it really opened a new window,
Yeah.
especially into these really intricate regulatory networks you find in organisms like cyanobacteria.
Okay.
NanoBiT allowed for just unprecedented quantitative precision, measuring PII's interaction with two key partners, one called PIPX.
PIPX.
Yeah, it's a transcriptional coactivator, helps control genes.
Okay.
And the other is an enzyme, N-acetyl L-glutamate kinase, or NAGK,
NAGK.
involved in making arginine.
That's the one. Crucial enzyme in arginine synthesis. Okay, so PII talks to both PIPX and NAGK. What did NanoBiT show? Let's start with, say, PII and PIPX, and the cell's energy state.
Right. So using NanoBiT, researchers could precisely measure how sensitive that PII-PIPX interaction is to the ratio of ATP versus ADP.
The energy balance.
Exactly. The assay showed a dissociation constant, the KD, which measures binding strength, lowers titer,
Right.
of 5.7 nanomolar when ADP was present. That's really strong binding.
Okay.
But with AGP, the KD jumped way up to 52.4 nanomolar.
Much weaker, like tenfold weaker.
Roughly, yeah. And the big insight here isn't just the numbers themselves. It's that NanoBiT's precision showed, like unequivocally, that ADP is the dominant effector,
Wow.
making this interaction a direct and really exquisitely sensitive readout of the cell's energy state.
And that matches what they see biologically.
Perfectly. It matches physiological observations, how ATP, p-ADP levels fluctuate when cyanobacteria go through light-dark shifts or when nutrients change.
Wow. That level of detail for something so fundamental like the cell's battery gauge, basically?
Pretty much.
Okay, what about the other player, 2-oxyglutarate, the carbon-nitrogen signal? How does that fit in?
Right. 2OG.
Yeah.
Crucial signal. NanoBiT accurately showed that 2OG gradually inhibits, it stops, the PI-PIPX complex from forming.
Okay.
And it pinned down the IC50, the concentration needed to inhibit, by half at 25 micromolar.
Right.
But what's even more revealing, especially for understanding how cells make decisions in real time,
Yeah.
is that NanoBiT could track the kinetics, the speed. So if ATP is present, 2OG makes the PII-PIPX complex fall apart really quickly. About 2 minutes.
Good S response.
Very fast. However, if ADP is also hanging around,
The low energy signal.
right, then that dissociation, that falling apart, is strongly mitigated. It takes much longer, like 7 to 8 times longer, roughly 340 seconds for half of it to fall apart.
Whoa. So the energy state dramatically changes how the cell reacts to the nutrient signal.
Precisely. It highlights this really sophisticated dynamic interplay. It's not just on-off.
It's nuanced. That paints a much more complex picture than I imagined. What about PII's other partner, NAGK, the enzyme?
Okay, PII-NAGGK. NanoBit confirmed they bind strongly when there are no effectors around or if ATP is there. Gave a KD of 8.8 nanomolar.
Tight binding.
Very tight. But crucially, it was also able to quantitatively measure a weaker interaction when ADP was present.
Ah, the one SPR struggled with.
Exactly. NanoBit measured a KD of 30.8 nanomolar. Weaker, yes, but still significant binding. And it confirmed that 2OG also inhibits PII neg-EK formation with an IC50 that matched previous findings pretty well.
Okay.
But the real kind of eye-opener came when they looked at what happens when both PipX and neg-EK are there at the same time.
Ah, the competition. The cellular tug-of-war.
Exactly. What happens when they're both competing for PII?
What did NanoBit reveal about that?
This is where measuring it all in solution, not stuck to a surface, really shines.
Right.
NanoBit showed that if ADP is present, the low-energy signal PII has a much higher affinity for PipX.
Prefers PipX.
Strongly prefers PipX, to the point where neg-EK basically cannot effectively compete it away.
Okay. Prioritizing gene regulation, maybe?
It seems like it. But if ATP is present high energy, the situation gets more complex.
How so?
Well, PII's affinity for neg-EK goes up, as we saw. But PipX still shows this remarkable staying power. It partially resists being kicked off even when there's a large excess of neg-EK around.
So it's not a simple switch even then?
Not at all. It really illustrates the cell's intricate balancing act. Low energy. Commit strongly to gene regulation via PipX. High energy. Okay, balance between metabolic regulation via neg-EK and gene expression via PipX.
That's fascinating. A real-time glimpse into the cell's internal resource allocation decisions. Now, you mentioned earlier NanoBit is great for those weak and transient interactions. Did that play out with PII?
Absolutely, and this was a genuine aha moment for the researchers involved.
Oh, yeah.
NanoBit was uniquely able to detect these sort of residual, very low affinity interactions between a specific PII variant called PIIS49E and neg-EK.
PIIS49E, what's special about that one?
It's a phosphamimetic. It's engineered to chemically mimic the phosphorylated state of PII.
Ah, like when PII gets a phosphate group attached.
Exactly.
Yeah.
Which usually happens under nitrogen-poor conditions, and it's generally thought to make PII largely let go of its partners, dissociate.
Okay, so you wouldn't expect much interaction.
Right. And older methods like SPR or BLI, they couldn't reliably detect any interaction there. It was below their limits.
Well, NanoBiT.
But NanoBiT clearly showed that even in this dissociated phosphorylated mimic state, a weak association still persists.
Really? So they don't completely separate?
Apparently not. They seem to maintain this sort of loose connection.
What does that imply then?
Well, this hidden interaction suggests that if conditions suddenly change back,
Right.
the complex could reform really quickly. They wouldn't need to find each other from scratch. It allows for potentially much more rapid cellular responses.
Keeping them on standby. Clever.
Very clever. And the studies also looked at other molecules like arginine and N-acetylglutamate, AG.
The product and substrate of NAG.
Right. Arginine, the product, it slightly reduces PII's affinity for neg-EK by about twofold. Okay.
Makes sense. Feedback inhibition.
Kind of. But here's the really surprising bit. NAG, the substrate of the enzyme,
Yeah.
it dramatically increases the PII-NAGK interaction, even when ADP is present.
Increases it. That's weird. Usually more substrate just gets processed.
Right. This suggests this unique feed-forward activation.
Feed forward.
Yeah, where having more substrate actually strengthens the enzyme regulator complex, directly boosting the enzyme's activity.
Yeah.
A very unusual finding.
Wow. Okay, so we've seen NanoBit really shine a light on the subtle dance of these metabolic regulators. Let's pivot now to an equally vital area, how cells interact with their environment
Right.
and how these interactions affect health and disease, specifically something called membrane protein trafficking. Why is the movement of these proteins so critical?
Well, membrane proteins, they're essentially the cell's gatekeepers and communication hubs on its surface.
The interface with the outside world?
Exactly. They're embedded in the cell's outer membrane, and they're involved in pretty much everything external. Sensing signals, cell communication, getting nutrients in, getting waste out. They're trafficking, which is this precise dynamic process of how they move to the cell surface, away from the cell surface, or even within the cell. It is fundamentally linked to a huge range of diseases.
Like what?
Oh, neurological disorders like hyperaccomplexia, certain types of epilepsy, cardiovascular issues like hypertension, even things like how viruses, think SARS-CoV-2, actually get into our cells. They often hijack these very receptors and their trafficking pathways.
So understanding this movement is key, and I'm guessing traditional methods for tracking this were also maybe falling short.
You guessed right. Precisely. Many existing ways to watch membrane proteins internalize, move into the cell, they had significant drawbacks.
Such as?
Some were really expensive. Others could only monitor very specific types of internalization mechanisms, not giving you the whole picture. Or they needed really specialized pricey equipment like gamma counters for radioactivity or high-end confocal microscopes for fluorescence.
Right.
NanoBiT came along and offered a new way. Highly sensitive, yes, but also remarkably versatile.
Okay, so how did they adapt NanoBiT for this? It sounds different from just seeing two proteins touch. How do you track movement into the cell?
The strategy here was really quite ingenious.
Okay.
So the researchers fused the small BiT fragment.
The little one.
Yeah, the little one. They attached it to the C-terminus, the tail end, of the membrane receptor itself, the protein whose journey they wanted to follow.
Okay, so the receptor carries the small bit.
Right. Then they took the large bit fragment and fused that to a different protein called the FYVE domain.
FYVE domain, what's that do?
The FYVE domain is like a well-known cellular address label. It specifically seeks out and binds to a particular lipid, phosphatidylinositol 3-phosphate,
Yeah.
or PI3P,
Okay, and where is PI3P found?
predominantly on the surface of early endosomes.
Early endosomes.
Yep.
Those are like the receiving stations inside the cell.
Exactly. They're the initial sorting hubs where proteins land right after they've been pulled in, internalized from the cell surface.
I see. So the receptor with small bit gets internalized. It reaches an early endosome where the FYVE domain with large bit is waiting. The small bit and large bit come together,
Can you get that beautiful, quantifiable luminescence signal?
indicating internalization just happened. That is brilliant.
It really is quite clever.
So what did they actually see with this system? Let's start with GPCRs, G protein-coupled receptors. They're huge drug targets, right?
Absolutely massive targets. And with GPCRs, like the well-studied D2 adrenergic receptor,
Okay.
NanoBiT allowed them to watch in real time both the internalization and the subsequent recycling back to the surface.
So you could see both.
Yes. For instance, they'd add an agonist like epinephrine to trigger internalization. And they'd see this rapid increase in the light signal as the receptor moved inside, usually peaking around 30 minutes.
Okay.
Then, if they watched the drug away, the signal would gradually decrease as the receptor recycled back to the cell surface.
Giving you kinetics for recycling, too.
Exactly. It provided crucial kinetic data, showing, for example, that recycling was a bit slower than internalization for this receptor. And they could do precise dose-response curves and double-check the signal was real using known drugs that block endocytosis.
Confirm the pathway.
Confirm the pathway, yeah.
And you mentioned seeing things earlier. Could you actually visualize this happening in live cells, not just get a number?
Yes, and that's another really cool feature. Using a bioluminescent microscope,
Okay.
they could actually visualize the B2 receptor trafficking in real time. It looked like these small, distinct spots of light moving dynamically through the cell's interior, the cytosol.
Wow.
That gives you this unique spatiotemporal insight, where and when it's happening. It's not so easy to get with other methods.
So it's not just for GPCRs. You said it was versatile. What else could NanoBiT tackle in terms of membrane protein movement?
Oh, yeah. Its versatility is pretty astounding. They successfully applied it to non-GPCRs, too, like the HER2 receptor.
HER2 involved in breast cancer.
Breast and ovarian cancers. Yes, crucial target. Its internalization was a bit slower than the GPCRs they looked at, but the method worked perfectly to monitor its trafficking.
Okay.
And maybe even more compelling, NanoBiT was used to study antibody-mediated internalization.
Antibodies making receptors go inside. Why is that important?
It's absolutely critical for developing new targeted cancer therapies. Things like antibody-drug conjugates, ADCs.
Ah, the magic bullets.
Kind of, yeah. The idea is you attach a potent drug payload to an antibody that specifically targets a receptor on cancer cells like HER2. The antibody binds, triggers internalization, and delivers the drug right inside the cancer cell.
So NanoBiT can watch that delivery happen.
It can track the efficiency and kinetics of that antibody-driven internalization process. Hugely valuable for developing better ADCs.
That's a direct line to new treatments. Okay, what about something that affects, well, pretty much everyone potentially, like viruses? Can this tech help us understand how they get in?
Right. This raises the really important question. Can we use this to watch viral entry? And the answer seems to be a definite yes.
How?
They actually developed a system specifically to monitor SARS-CoV-2 viral entry.
The COVID virus.
Exactly. They adapted the SARS-CoV-2 spike protein, the part that binds to our cells, within a safe, lab-based lentiviral system.
Okay.
And then in the cells, they express the ACE2 receptor, which is the main doorway for the virus.
Right.
But they linked ACE2 to the small bit,
Ah, setting the trap.
pretty much. So they could then observe the spike protein binding to ACE2 and the subsequent internalization of the virus into the cell, again, via those early endobounds where the large bit FYVE was waiting.
And what did they see? Was it fast? Slow?
Interestingly, this whole process was much slower than when you trigger receptor internalization with a drug.
Really?
Yeah, it took approximately 3 hours for the entire infection process to register the full light signal.
3 hours? That's quite a while.
It is, which gives you an idea of the complexity of viral entry compared to simple receptor internalization. But the key thing is this capability. It opens up huge possibilities,
Like what?
like screening for neutralizing antibodies, antibodies that block entry, or testing antiviral drugs that target the entry process. You could do it in real time, in a relevant cellular context. Could dramatically speed up finding new therapies.
So, taking a step back, what does all this mean for us, for drug discovery, for just understanding life? What's the big picture here?
Well, if we connect all these dots, NanoBiT really provides this powerful and remarkably universal methodology.
Reversal.
Yeah, the ability to monitor such a wide range of internalization mechanisms, GPCRs, other receptors like RTKs, the action therapeutic antibodies, even complex viral entry pathways,
Right.
all in real time and, crucially, in living systems. That profoundly accelerates drug discovery and development for just countless diseases where this membrane protein trafficking is a key player.
It's like turning the lights on in a previously dark room.
Exactly. It gives us this unprecedented dynamic window into these absolutely critical cellular processes. We're moving away from just static snapshots
Yeah.
towards watching real-time movies of life's fundamental machinery in action.
This whole deep dive into NanoBit, it honestly feels like we've been given a kind of superpower. The ability to shine a light on these incredibly intricate, hidden communications happening inside our cells, really revealing the choreography of proteins.
It is quite profound.
Yeah, from sensing tiny metabolic shifts to tracking how viruses invade, the insights are just, profound.
Indeed. And I think the capacity to quantitatively analyze these interactions, especially with such high sensitivity,
Right.
particularly those weak transient ones that were basically invisible before, that fundamentally changes how we can even begin to model and understand the truly complex networks that govern biological systems.
Moving beyond simple diagrams.
Absolutely. It's about getting that dynamic, that holistic view of how cells actually function moment to moment.
And that leads us perfectly to our final provocative thought for you listening. Given NanoBT's unique power, its ability to reveal even the most fleeting, the most transient protein interactions, what previously undetectable biological mechanisms, things we couldn't even see before, might we now finally be able to observe and really understand? And could that unlock completely new avenues for treatment or maybe even biological control simply because we can finally see them?